Welcome to the differential gene expression module, and in this talk we're going to look at DNA and histones. In this talk we're going to start with the central dogma followed by the postulates of differential gene expression. We'll then look at the different ways of regulating gene expression as well as different aspects of DNA packaging, which includes the formation of a nucleosome and the role of histone H1. We will also learn about the differences between heterochromatin and euchromatin and how we can change heterochromatin to euchromatin and vice versa using different histone modifications. We will also learn about the different enzymes that mediate histone modifications and what is the histone code. The central dogma helps us understand how DNA that is present in the cells is able to code for information that allows proteins to be formed. Now, the steps involved from DNA to protein is seen in all living organisms, and that is why this is called as the central dogma. To start with, the central dogma states that the DNA that is present in cells of all organisms can be duplicated by the process of DNA replication. In addition to DNA replication, we can have DNA repair and exchange of DNA through genetic recombination. But in all instances, DNA replication happens. The DNA can then be transcribed to RNA, and this process is called as transcription. Thus, the synthesis of RNA from DNA is transcription. However, it should be noted that viruses have the ability of making DNA from RNA, and that process is called as reverse transcription. Now, once the RNA is made, the RNA that has information from the DNA is able to form proteins through the process of translation. Thus, according to the central dogma, the DNA is able to code for proteins by being transcribed first to form RNA, which then gets translated to form proteins. Now, differential gene expression is a concept that helps us to explain how the cells in the same organism are different from one another. An explanation that allows us to understand why cells of the same organism are different is due to the expression of a unique combination of genes in the cells of the body. There are certain postulates of differential gene expression, and the first one states that every somatic cell nucleus of an organism is going to contain the complete genome that was established when the egg was fertilized by a sperm. And what this basically states is that the DNA of all differentiated cells in an organism is identical. The unused genes in differentiated cells are neither destroyed nor mutated. So every cell in the organism has genes that are not necessarily used by the cell. And when that happens, those genes are retained and they have the potential for being expressed. Another postulate is that only a small percentage of the genome is expressed in each cell of the organism, and a portion of the RNA that is synthesized in each cell is specific for that cell type.
This brings us to the concept of regulation of gene expression, which basically means that there has to be ways to determine what genes are expressed and which ones are not. So the way regulation of gene expression can occur is through differential gene transcription. This process regulates which of the nuclear genes are going to be transcribed into nuclear RNA. Another way of regulating gene expression is selective nuclear RNA processing. In this process, it is regulated which of the transcribed RNAs are able to enter into the cytoplasm and become messenger RNAs. It should be noted that in eukaryotic cells, transcription occurs in the nucleus, However, the RNA that is transcribed has to be transported to the cytoplasm in order for it to be translated into a protein. Another mechanism of regulating gene expression is selective messenger RNA translation. In this process, the type of messenger RNAs present in the cytoplasm that have to be translated into proteins is regulated. Or in other words, not every mRNA present in the cytoplasm is translated. The cell can control which ones should be translated and which ones should not. Finally, another way of regulating gene expression is differential protein modification. A protein can sometimes have to be modified in order to remain functional, and hence we need these protein modifications, and this is a way of regulating protein function. We're now going to look at how to obtain access to genes. As we all know, genes are sequences of DNA that code for proteins. In the case of eukaryotic cells, the DNA is associated with proteins to form what we call as chromatin. The most abundant protein present in chromatin are histones. The DNA is wrapped around a histone octamer to form a structure that's called as a nucleosome, as we can see in the figure that is shown below. Here we can see two nucleosome structures as depicted in the blue box. In each nucleosome, there are 8 histone molecules, which is why it's called as an octamer, and there are 2 H2A, 2 H2B, 2 H3, and 2 H4 histone molecules. About 147 base pairs of DNA are wrapped around this histone octamer to form as a nucleosome, and hence it resembles a bead that is present with string wrapped around it. In addition to the histones that we described previously, there's another histone that's also present in chromatin called Histone H1. This one binds to the nucleosome and the DNA and alters the path of the DNA to allow the DNA to be more compact. So usually for every nucleosome, there's only one histone H1 that is present. As shown in the figure below, we have a nucleosome that is depicted by the yellow round circle, and the histone H1 is shown in the form of that green boomerang structure. The histone H1 interacts with the DNA and the histone octamer, and it changes the path of the DNA as shown in the figure to the right, where now we can see a zigzag structure of the nucleosomes that are now close to each other, and this allows the DNA to become more compact. In the case of cells, the DNA has to be present in a very compact structure so that it can fit into the small size of the nucleus. And hence in this figure, we are looking at different steps that are involved in packaging. While you don't have to remember all these steps, what you should remember is that of the nucleum or the beads on the string form of chromatin, which is the second form shown on the left part of the figure, as well as the solenoid structure. After that, there are many more steps for packaging, but we don't have to remember these different steps. For genes to be transcribed, the chromatin must be accessible to the different proteins that play a role in gene transcription. When the chromatin is loosely packed, as shown in the figure below, especially the portion in the blue box, then that kind of chromatin is called as euchromatin. Now, as you can see in that figure, the small red boxes show sequences of DNA that are available or accessible. And thus, if there are genes in these regions, then these genes are also accessible to various proteins that are involved in transcription and hence will be transcribed. On the other hand, if the chromatin is tightly packed, which is what is shown on the right side of the figure, then such chromatin is called heterochromatin. In the case of heterochromatin, the genes are not accessible because, as we can see in the figure, there are no DNA sequences that are open, as we saw in the case of euchromatin. And hence, in heterochromatin, genes are not usually transcribed. Now, gene transcription can be regulated by controlling the packaging of chromatin. To control the packaging of chromatin, the histones can actually be covalently modified. The histones have these N-terminal tails that are extending out of the nucleosome, as can be shown in the figure below. If we look at the figure below, all the parts in these blue boxes are basically the N-terminal tails of the histones that are extending out of the nucleosome and are normally covalently modified. The most common covalent modifications observed in histones are acetylation, deacetylation, methylation, and demethylation. So let's have a quick look at what happens in these modifications. In the case of histone acetylation, an acetyl group is basically attached to the N-terminal tails of some of the histones. The acetylation is mediated by histone acetyltransferases, or HATs. Acetylation loosens histones and activates transcription because now the histones are not associated with each other as tightly, and thus, as we can see in the figure below, histone acetylation causes the nucleosomes to remain farther from each other. These acetyl groups can be removed by histone deacetylases that are also called as HDACs, and the removal of the histones stabilizes the nucleosomes so that they can interact with each other quite strongly, as shown in the figure to the left, and this results in heterochromatin, and thus the genes are not accessible anymore. In the case of histone methylation, a methyl group is added to certain amino acids present on the histones. The methylation is mediated by the enzymes histone methyltransferases, or HMTs, and is usually associated with repression of transcription, basically, which means that transcription does not occur. However, in some cases, depending on the presence of neighboring acetyl or methyl groups, methylation can actually cause the activation of gene expression. Methyl groups can be removed by the group family of enzymes, which are the histone demethylases. It's good to know that there is a concept now emerging of the histone code. The histone code basically means that there are combinations of covalent modifications on a histone that can signal very specific outcomes. To give you an example, when we look at the figure part A, which is at the top, we're looking at one of the histones, which is histone H3, and we're looking at the different modifications that can happen in the different amino acids that are denoted by the letters R, K, S. R stands for arginine, K stands for lysine, and S stands for serine. When we look at the circles that have a color associated with them, M stands for methylation, A stands for acetylation, and P stands for phosphorylation. So in figure A, we are shown the possible covalent modifications that can happen in histone H3 and the location of the amino acids where these modifications can occur. In figure B, what we're looking at is the different modifications that can happen and what is the outcome of that modification. For example, if there is 3 methyl groups that are added to the lysine 9, then it is normally associated with heterochromatin formation or gene silencing. However, if there are 3 methyl groups on lysine 4 and 1 acetyl group on lysine 9, as shown in the middle figure of the B panel, it is associated with gene expression. Finally, the last one shows that if there are 3 methyl groups on the lysine 27 of histone H3, it is associated with gene silencing. Thus, the covalent modifications can recruit other protein and protein complexes whose actions can result in a biological response. It is not necessary for you to remember the different combinations of the histone code. It's enough to know that there is such a concept called as the histone code. There are ways for a cell to maintain the memory of modifications that have been done on histones. The modifications of histones can recruit proteins that retain the memory of the transcriptional state across different cell generations. We're going to look at 2 special family members that are involved in retaining the memory of these modifications. The first family are the polycomb family of proteins, and they retain a memory of repression. The polycomb family of proteins, they methylate lysines on H3K7 and H3K9 to repress gene activity. Other polycomb factors can bind to methylated histone 3 or H3 and methylate adjacent nucleosomes to form heterochromatin. Another family of proteins that plays a role in retaining the memory of histone modifications are the trithorax family, and they tend to retain the memory of activation of transcription. Thus, the polycomb family retains the memory of repression of transcription, while the trithorax family retains the memory of activation of transcription.
With this, we come to the end of our talk, where we looked at the central dogma and postulates of differential gene expression. We also looked at different ways by which gene expression can be regulated and how DNA is packaged to form heterochromatin and euchromatin. We learned how histone modifications are able to convert heterochromatin into euchromatin and vice versa, and the various enzymes that mediate histone modifications. We learned about the concept of the histone code and how the histone code can sometimes help in retaining the memory of the transcriptional state of genes across different cell generations.